This recording is going to cover the secretions and the functions of the secretions from accessory organs that are important in digestion, and specifically the accessory organs of the liver, the gallbladder, and the pancreas. The secretions from these organs are going to mix with the chyme from the stomach in the small intestine, and secretions from the small intestine will also obviously aid in digestion. So specifically, we're just going to look at pancreas and liver and gallbladder. So the pancreas, you can see the outline right here. That is a major, it's actually an endocrine as well as an exocrine organ. So we've talked about its endocrine function, that it produces such things as insulin and glucagon, but most of that organ has an exocrine function. So the contents from the pancreas will empty into the duodenum right here, and it's through an opening called the ampulla of Vater, or the hepatopancreatic ampulla. It will mix with secretions from the gallbladder. In the gallbladder, it will be storing bile, and that will go down through the common bile duct at some point. Here it's a cystic duct, and bile needs to empty again into the duodenum, plus any bile that had come from the liver. Now, liver produces bile, and it goes to the right and left hepatic ducts, and it can be stored up here in the gallbladder or come down here and contribute to the secretions there. In my case, I don't have this, and so my bile is directly coming from the liver. So let's just look at that pancreas. So pancreas has, I mentioned, that hepatopancreatic ampulla, or the ampulla of Vater. Here you actually see the hepatopancreatic sphincter. The hepatopancreatic sphincter is also called the sphincter of Oddi. That needs to be relaxed in order for the bile from the common bile duct, as well as the enzymes and bicarbonate from the pancreatic duct, empty in the duodenum. Now, what are some of the things that the pancreas secrete? Well, let's go through it. So the exocrine pancreas, you have these acinar cells. The acinar cells produce a number of different enzymes. Amylase digests starch. So that is involved in the digestion of starch. We do have amylase that's secreted by the salivary glands, but most of us do not chew our food enough or long enough that that's going to be completely break down that starch into, and actually the lowest thing that amylase really can digest starch into is maltose. So we need to add some more amylase into our food. Lipases break down fats, and then we have these enzymes that we refer to as proteases. Well, these are all proteolytic enzymes, and they're stored in the pancreas as an inactive form. The trypsinogen is inactive. Where you have it in gold, that's the active form of the enzyme. So trypsinogen has to be converted to trypsin before it can start digesting proteins. Chymotrypsinogen has to be converted to chymotrypsin. Procarboxypeptidase has to be converted to carboxypeptidase, and proelastase, which actually breaks down elastin fibers, that has to be converted to elastase. Well, the reason why in the pancreas these proteolytic enzymes are produced and stored in an inactive form is that these, if they become activated, they will digest the pancreas. And trust me, it's extremely painful. When I had my gallbladder issue, is I also had a pancreatic issue. I had cholecystitis as well as pancreatitis, and it was the pancreatitis that would knock me on my butt because it was so painful. It was starting to digest my pancreas. So I end up having, I mean, the pancreas hopefully is fine now. I had to end up having my gallbladder removed. So you don't want these enzymes activated in the pancreas because it'll digest your pancreas. So they need to be activated while they're in the small intestine. Now we're going to show you how they're going to become activated. Now, in a moment. The cells that line the pancreatic duct, or the ductal cells, they produce bicarbonate. That bicarbonate is extremely important. The bicarbonate will help to neutralize that acidic chyme, providing the proper pH for enzymatic digestion. Plus, also, you don't want the chyme that its acid would damage the mucosal lining of that small intestine. Now, those proteolytic enzymes, or proteases or proteinases, it's all the same thing. Those will become activated hopefully, once they're in the duodenum. What happens is there is a enzyme located in your duodenum called enterokinase. That's a secretion from cells of the small intestine. That will convert trypsinogen to trypsin. Trypsin then can activate everybody else. So you have to activate trypsinogen first, and then it activates everybody else. Now you'll see this term, pancreatic zymogens. That's the term for these here that are in an inactive form. They're called zymogen granules. They're inactive. They have to be converted to an active form. So what happens is you cut off a few amino acids, the protein will kind of fold up and assume its proper 3D conformation, and then it becomes active. And so this should not happen until you're in the duodenum. The enterokinase is, you notice it says brush border, is secreted by the brush border, just represents the microvilli that you have lining the small intestine. The bicarbonate, I had just mentioned, is secreted by the ductal cells. You don't have to sit there and memorize this right here, but the bicarbonate is secreted into that pancreatic duct, and I had mentioned why is it necessary. You need to have proper pH for enzymatic action, because the enzymes that are being secreted by the small intestine as well as the pancreas do not work well at acidic pH. Also, it helps to protect the mucosal lining, okay, because the acid would damage it. The regulation of pancreatic secretions. There's a number of things that regulate it. Two major things are hormones. So let's look at the hormones first. Secretin and cholecystokinin. Cholecystokinin is abbreviated as CCK. Perfectly acceptable to abbreviate that hormone as CCK. Both secretin and cholecystokinin are hormones produced by the small intestine. These are involved in regulating digestion. Well, we'll do secretin first. Secretin stimulates the production of the bicarbonate from the ductile cells. A potent stimulus for the secretion of secretin, which then goes into the bloodstream, then targets that pancreas, is acid. So acid in the chyme. So it's like, well, a lot of acid, I need more bicarbonate. Secretin's going to handle it. Cholecystokinin stimulates the production and secretion of the pancreatic enzymes. So a potent stimulus for CCK to be secreted by the small intestinal cells is actually the presence of proteins or protein degradation products in the chyme and fats. And that's going to become important later, something else. But so remember, the pancreas secretes lipases, which help with the digestion of fats, and it secretes all those proteinases, which help with the digestion of protein. So those are potent stimuli for more cholecystokinin to be secreted. So you've got hormones that stimulate secretion of things from the pancreas. You also have the role of the autonomic nervous system. So the vagus nerve, very important nerve that has a lot of parasympathetic fibers. That increases secretion of should say the enzymes and bicarbonate from the pancreas. But it's relatively mild. It's more important are secretin and cholecystokinin, but that does stimulate secretions. The sympathetics opposite. Sympathetics not rest and digest. That actually decreases pancreatic secretion. Okay. Now the liver is the next thing. We're going to do liver and then talk about gallbladder because they're kind of very closely related. And what they're going to deal with is the liver is where we produce bile. And the hepatocytes in the liver produce the bile. The bile will travel via the right or left hepatic ducts, enter into the common hepatic duct, and in between meals, usually it's going to come up here and be stored in the gallbladder. And in the gallbladder, the gallbladder will help to kind of concentrate it. Well, what's the function of the bile? So let's talk about the function of bile. Let's look at the composition. So you got water in it. These two things, the important component of bile that we need to think about. The bile salts and lecithin, they act as emulsifiers. They emulsify fat. They do not break the bonds in fats, but they emulsify them. And I'm going to show you a picture to show you that. They increase the surface area so lipases can work better. They do also aid in the absorption of fatty acids once you've kind of broken them down, and also aids in absorption of things that are fat soluble, or like the fat soluble vitamins like A, E, D, and K. So they kind of grab a hold of it and allow these things that are fat soluble to increases their absorption of it. Now, bile does have some pigments, gives it some color to it. It has some cholesterol, and it also has obviously some electrolytes to it. But let's look at its role as the bile salts and lecithin as emulsification. So emulsification of fat. So you have this big old fat globule. The fat wants to hang with other fat. It doesn't. It's water fearing. Doesn't like to hang with water. So what we need to do is the bile salts and lecithin too, they are, we call amphipathic. They have a polar end, which is hydrophilic, and they have a nonpolar end, which is hydrophobic. And what it does is it orients itself around the little fat droplets, in which the nonpolar side, the hydrophobic side, is hanging around with the fat, and then the hydrophilic end is out here in the aqueous environment where the water is. So what it's doing is preventing all these fat droplets to kind of congregate together like this. It's breaking them up into smaller pieces. The fat is still fat. You have glycerol plus 3 fatty acids in a triglyceride. It does not break the bonds in that, but all it's doing is keeping one fat molecule from hanging out with another fat. And so we've increased the surface area so lipases can access that and break these bonds. So you can get your 3 fatty acids released from the glycerol backbone. So the emulsification of fat is a mechanical, not a biochemical means of digestion. So it does not break the bonds in that actual fat. So it aids in the digestion of fat. Actually, I'm going to just go on this because I need a kind of a blank page. I'm going to give you an example why it's important, or give you a little experiment. If we take a test tube and I throw in fat, so I'll put fat right here. I'm going to throw some bile. I'll just do bile actually I should do bile as green because it is greenish. So throw some bile in there and I shake it up, but I incubate it at proper temperature, all that. And I want to see if fat was digested. And technically what you can do is, this was a real experiment, is if fat's digested, you're going to get fatty acids. And you can actually measure the pH. And if you've got digestion, the pH would drop. So I'm going to check what happens with test tube A. Now test tube B is going to have fat in it. It's going to have lipases in it. So that's test tube B. Test tube C is going to have fat in it, bile salts, and lipases. If I look at the results from test tube A, I'm not going to see any change in pH. Bile salts don't break the chemical bonds in it. I would see no change in pH. If I looked at the test tube with fat and lipases, I would see a slight decrease in pH because I need to increase that surface area of the fat because the fat's going to stick to each other. It's going to be hard for those lipases to get in there. So if I add some bile salts, I'm going to see that pH drop the greatest. I'll have the best digestion in that tube. So the bile salts make it better for the lipases to work. Okay, so that's a good thing. So what regulates bile release? Well, first thing we got to just mention again, we got the bile. It's stored in the gallbladder if you've got one. The gallbladder, what's going to happen in the gallbladder, it helps to concentrate that bile. And so you've eaten a meal. If you need the bile, because they help the digestion of fat, you want the gallbladder to contract because it's got smooth muscle associated with it. So it contracts, the bile should come down here, and then you want this hepatopancreatic sphincter, or that sphincter of Oddi, to relax so the bile can be released into the duodenum. So what regulates it? Okay, so you have chyme from the stomach, we're going to say there's some fat in there, you had some fat with your meal, is the cells in the duodenum secrete cholecystokinin, which I've mentioned previously. And I said a potent stimulus for the release of CCK is the presence of proteins as well as fat. So cholecystokinin will go into the bloodstream, and besides what it does to the pancreas, it stimulates contractions of the gallbladder and relaxes that sphincter of Oddi. And so we can get that bile into the duodenum to help aid in the digestion of fat. Plus, relaxing that sphincter of Oddi also allows the lipases from the pancreas to empty into the small intestine. So that's a major thing for the regulation of bile, is that. Now, if you do not have a gallbladder, what's going to happen? You're still going to have that, you ate some fat in your meal. You're still going to have CCK being released. But instead of the contraction of the gallbladder, which you don't have, you still will relax this sphincter because you still make bile in the liver. And so that bile that's coming from the liver is going to be released in there. The only problem is that bile is less concentrated. And so people who lack a gallbladder, not everyone, but I'm one of them, I have to watch the amount of fat I take in at one sitting because the bile that's being released into my duodenum is not as concentrated. And so the digestion of fat is not going to be as effective, and that will irritate my intestines, and it causes discomfort. Just put it that, cause a little discomfort. slightly painful. So it's also actually good way of keeping yourself from eating too much fat at one meal. All right, so that's where we're going to stop. So next recording we're going to look at the small intestine.